Okay, so our very last section of chapter 10 covering photosynthesis is looking at the Calvin cycle. The Calvin cycle is what we call the dark reactions. They don't depend on the presence of light. However, as we've already covered when we looked at the light reactions, our Calvin cycle depends on the ATP and the NADPH that are produced in the light cycles. So since these are very unstable molecules, the Calvin cycle still happens during the daytime, but they don't operate directly off of the light. They operate off of the ATP and NADPH produced by the two photosystems, photosystem 1 and photosystems 2. Now, the Calvin cycle is the reaction that produces our carbohydrates, our sugars, from carbon dioxide. And they are also known as a light independent reaction. So we have to have the ATP and the NADPH, these high energy molecules produced by the light dependent reactions. And the Calvin cycle is actually going to use carbon fixation. What they're going to do is they're going to use that carbon dioxide and they are going to fixate or attach an organic compound to that. Okay, so when they were looking at how all of this happens, when they were looking at the Calvin cycle, they wanted to know what intermediates are reduced. Carbon dioxide is reduced to sugar. Now remember, glycolysis had 10 steps in it. So we're basically working backwards to glycolysis in some degrees. So there's going to be some intermediates between carbon dioxide and our carbohydrate or our sugar. So what they did is they added a radioactive carbon dioxide to actively photosynthesizing algae. And then they waited a little while to see where the radioactive molecules would be seen. So they would wait in between 5 and 60 seconds, and then they would kill the cells by immersing them in hot alcohol, basically stopping the process of photosynthesis. And then they would separate the molecules using chromatography, and they would then lay an X-ray film on top of that chromatograph so that they could actually see the radioactive labels. What happens is when you lay X-ray film on radioactive material, it's going to show up as these dark spots that you see at the bottom here. So after 5 seconds, they saw that we had this one compound called 3-phosphoglycerate. Now this is actually one of the intermediates in glycolysis. So it makes sense that we would also find it in the Calvin cycle of photosynthesis. So what they concluded is that this was actually the first intermediate product, and then other intermediate products appear later, none of which we're really going to cover in detail. Now what you do need to know is that the Calvin cycle has 3 phases: fixation, reduction, and regeneration. Now typically the Calvin cycle is going to fixate a carbon dioxide with a 5-carbon compound. So this CO2 is going to react with a 5-carbon compound. This 5-carbon compound is known as ribulose biphosphate. We're just going to call it RUBP from now on, just to make it a little bit easier. But RUBP is a 5-carbon compound. So when we get this fixation reaction, we're going to add the carbon from the carbon dioxide and the 5 carbons from the 5-carbon compound, and we're going to get a 6 carbon molecule that is then going to split into two 3-carbon molecules of what is known as 3-PGA. So 3-PGA is the 6-carbon molecule that I've been talking about. These are going to be combined to form our glucose molecules. But this is a very simple carbohydrate. A 3-carbon molecule of 3-PGA is a very simple carbohydrate. Now, after the 3-PGA are used, we're then going to have reduction for the next phase. Basically, once we have these 3-PGAs, we can then phosphorylate them by using ATP, and we can reduce the 3-PGA with NADPH. So essentially, we're going to transfer the energy from the ATP and the NADPH to this 3-carbon molecule, this 3-PGA. And what that's going to do is that's going to produce glyceraldehyde 3-phosphate, or G3P. And some of that G3P is used to make sugar. So this is going to be really, really important to our Calvin cycle. So we have a 3-carbon molecule at the end of fixation. Now we have a 3-carbon sugar. Essentially, we have a 3-carbon carbohydrate that is going to be used to make sugars like glucose. Now, not all of the G3P is used to make sugars. It's actually a fairly small amount. Most of the G3P is actually used in reactions that use more ATP from our light-dependent reactions, which will regenerate this RUBP, which was found in the fixation stage. So most of the energy that we're producing in the Calvin cycle does not actually go towards making carbohydrates. It doesn't actually go towards making glucose. Most of the energy used in the Calvin cycle is used to further propel the carbon cycle, which is why it's called a cycle, is because that G3P, most of that is going to be used to regenerate the RUBP in the first fixation stage. So here's a breakdown of our 3 phases here. We have fixation, reduction, and regeneration. So the reaction occurs in a cycle. We really need quite a bit of input, you're going to see over here on the right side, in order for this cycle to occur. So we have 3 carbon dioxide molecules going in to the fixation here, which results in 3 PGA molecules. We're going to add 6 molecules of ATP. We're going to add 6 molecules of NADPH, which will reduce the 3-PGA to the G3P. This is going to be our building block for our sugars later on. And so one of these 3 molecules of G3P is going to be used to build glucose. However, the rest of those, the remaining, there's actually going to be 5 G3P, are going to be passed on to regenerate the RUBP, which is used during the fixation cycle, or the fixation phase of the cycle, excuse me. Okay, so we have over here our equations. So with fixation, we have RUBP and carbon dioxide that forms 3-PGA. We have our reduction, which takes the 3-PGA plus 6 ATP and 6 NADPH, and we get 1 G3P that continues on to make other organic molecules such as glucose, and 5 that are used to continue the Calvin cycle. So these 5 G3P basically use more ATP to regenerate the RUBP, which happens down here. So our 5 G3P goes to step 3. We add 3 more ATP, and we regenerate 3 RUBP. So we are generating the same amount that we are using up here in the fixation stage. Now, the Calvin cycle stroma of chloroplast. One turn of the cycle fixes 1 molecule of CO3. So we actually need 3 turns of the cycle to fix 3 carbon dioxide molecules, produce 1 molecule of G3P, and 3 regenerated RUBP. So when they discovered this Calvin cycle, and by the way, the scientist who led most of the discovery here, his last name was Calvin, which is why it's called the Calvin cycle. They looked at how ATP and NADPH are produced by the light capturing reactions and how these allow the cell to reduce carbon dioxide to a carbohydrate. Now, in doing this, we need something that helps speed up the process to make it more efficient. So we actually discovered this enzyme, a CO2 fixing enzyme that helps with this process. The CO2 fixing enzyme is called ribulose 1,5-biphosphate carboxylate / oxygenase. We're going to call it Rubisco. Rubisco is found in all photosynthetic organisms that use the Calvin cycle to fix carbon, because it is the carbon dioxide fixing enzyme. So if a photosynthetic organism uses carbon dioxide, they are going to use Rubisco as the enzyme. And it's thought to be the most abundant enzyme on Earth because most of our biomass has to be photosynthetic organisms to basically supply food for the rest of the food chain. This is what the Rubisco looks like. It is a massive enzyme with lots and lots of different parts to it. And you'll notice that it has several different active sites there that look like little yellow flowers. Now, Rubisco is a very slow enzyme compared to what we would normally think of enzymatic reactions. It's also very inefficient because it can catalyze the addition of either a CO2 or an oxygen molecule to that RUBP. If it catalyzes CO2 and RUBP, well, then we're driving the Calvin cycle, and that's what we want. When oxygen catalyzes with RUBP, it's not really producing the sugars that we need for the plant. However, they have found out that this isn't entirely useless, that when oxygen binds to RUBP, it does produce different products that are used for plant growth and development. So either oxygen or the carbon dioxide basically the oxygen and the carbon dioxide are going to compete with each other at the Rubisco's active sites. And if O2 binds to the active sites, then it's going to slow the rate of CO2 reduction. Therefore, it's going to slow the rate of sugar production. Okay, now when oxygen and RUBP react in the Rubisco's active site, one of the products that undergoes a process called photorespiration. Respiration is what we've already looked at. It's cellular respiration. We're using oxygen. So it makes sense that if plants are going to use oxygen, then they are going to undergo a process called photorespiration. And it basically undoes photosynthesis because it consumes oxygen and releases fixed carbon dioxide. However, again, we're gaining more and more knowledge about how this is actually beneficial for the plant. Photorespiration can be involved in plant signaling, and it may have a protective role as well. All right, so with our reactions for photosynthesis and photorespiration, we can kind of see the difference here. RUBP and CO2, this is photosynthesis. This specifically is in the Calvin cycle. We're going to get 2,3-phosphoglycerate out of this. When our RUBP reacts with oxygen, we're going to get the 3-phosphoglycerate, but we're also going to get a 2-phosphoglycerate. And when that is processed, CO2 is released and ATP is used, which essentially undoes photosynthesis. But again, this has a benefit for the plant. Now, photosynthesis is going to be regulated just like cellular respiration is. Light is going to trigger the production of photosynthesis proteins, and it's going to activate our enzyme Rubisco. When we have lots of sugar, just like when we have lots of ATP in cellular respiration, that high sugar is going to inhibit the production of photosynthetic proteins. However, we're still going to have the production of proteins that process and store the sugars, because even though we have a high amount of sugars, we need to make sure that they are going to be stored for a later time. Now, photosynthesis is also going to be inhibited with a low CO2. If we don't have very much CO2 available, then it's going to inhibit the Rubisco. Because if there's low CO2, there's often a high amount of oxygen, and we don't want the oxygen to bind to the active site of the Rubisco because we don't want to undo the photosynthesis. Carbon fixation is favored over photorespiration when concentrations of CO2 are higher than oxygen. So if we have a low CO2, we want to shut down the Rubisco so that we don't have too much photorespiration, because we don't want to undo the photosynthesis. Now, we often have a problem with how plants get their carbon dioxide. Normally we think, oh, well, they just breathe the oxygen and the carbon dioxide just diffuse down their concentration gradients. However, plants are covered with a waxy coating, which doesn't allow CO2 in and doesn't allow oxygen out. This is beneficial for the plant because the waxy coating actually prevents water from evaporating. But unfortunately, it also prevents the transport of carbon dioxide and oxygen down their concentration gradient. So plants have these pores called stromata that allow gas exchange. They look like little mouths in a leaf. They're basically controlled by two guard cells that change the shape, that either open or close the stroma or the stromata. Here's what they look like. At the top diagram here, we see leaf surfaces that contain stromata there. All right, now those open stromata allow the CO2 to now diffuse into the leaf and for oxygen to diffuse out. And so we're going to have a strong concentration gradient here favoring the entering of CO2 because that carbon dioxide is constantly being used up by the Calvin cycle. So there's always going to be a strong concentration gradient that allows carbon dioxide to move into the leaf through the stromata. These are usually open during the day and closed at night because this is when photosynthesis is happening. They need that CO2 during the day. However, when conditions are really hot and dry, plants adapt to that by closing their stromata. This prevents evaporation of water. When it's hot and dry and the stromata close, we're going to have an oxygen buildup in the leaf. And when we have an oxygen buildup, now we're going to have a situation that favors photorespiration because that oxygen is going to bind with our enzyme, and photorespiration is going to occur, undoing the photosynthesis. To kind of combat this, plants have adapted, or God has given plants the ability to have the C4 pathway. So with C4 plants, these are going to be things such as sugarcane and maize. And they're called C4 because they fix the carbon dioxide to a 3-carbon compound instead of a 5-carbon compound, which is going to result in a 4-carbon compound. So 1 carbon in our CO2, 3 carbons in our 3-carbon compound. This is going to result in a 4-carbon compound, which is why it's called a C4 pathway. Now, let's compare that to our Calvin cycle. Our Calvin cycle results in 2 3-carbon compounds. So we're going to call our Calvin cycle the C3 pathway. Now, the C4 pathway does not completely and totally take the place of the Calvin cycle or the C3 pathway. Instead, it occurs with the Calvin cycle. So it occurs in addition to the carbon fixation to RUBP by Rubisco. So here's an example here of the difference between our C4 plants and our C3 plants. In our C4 plants, we're going to have carbon dioxide attach or fix to a 3-carbon compound, resulting in a 4-carbon organic acid. And in our C3 plants, our typical Calvin cycle, our carbon dioxide is going to bind to our 5-carbon compound, our RUBP. It's going to be handled by our enzyme Rubisco, and we're going to get 2 3-carbon organic acid molecules. Now, even though these occur together, they occur in separate cells. So in C4 plants, carbon fixation and the Calvin cycle occur in separate cells. Basically, what happens here is we have our carbon dioxide fixation to our 3-carbon molecule in our mesophyll cells, which are near the surface, and that's going to form a 4-carbon molecules. 1 carbon for our CO2, 3 carbons for our molecule, resulting in a 4-carbon molecule. Okay, so this 4-carbon molecule that was formed in the mesophyll cells near the surface is then going to travel to bundle sheath cells. These are protected in the interior of the leaf, so they're just kind of basically pulled in to a separate room, if you will. In this separate area, those 4-carbon molecules are then broken down to release the carbon dioxide again, which is then used in the Calvin cycle. So this kind of prevents the issue of having too much oxygen around, which would result in photorespiration. We're essentially packaging up our carbon dioxide through the C4 processes, transporting them into a separate room, releasing the carbon dioxide where there is no oxygen, and now we're going to complete the Calvin cycle. So now and then once we're done releasing the carbon dioxide, our 3-carbon molecule is going to return to the mesophyll cells to fix more carbon dioxide. So here's our C4 plant. Our mesophyll cells near the surface is going to contain this PEP carboxylate. This is what is going to fix the carbon dioxide to a 3-carbon compound. And then the resulting 4-carbon compound is going to move into the vascular tissue, basically where those bundle sheaths are, so that the Calvin cycle can occur. And this basically eliminates the worry of having too much oxygen that would result in photorespiration. Now we also have these really unique plants that we call cam plants. Cam plants, an example is cactus. These are plants that live in really extreme hot and arid environments. So we really need to make sure that we limit the oxygen that is building up in the leaves. However, we also don't want to open up those stromata because we don't want to release the water that's in the leaves either. Cactus have a very waxy surface as to eliminate their loss of water. So these cam plants are also able to fix carbon dioxide to a 3-carbon compound in order to form a 4-carbon compound, just like our C4 plants. This again also occurs in addition to the regular Calvin cycle carbon fixation to our 5-carbon sugar, RUBP, by Rubisco. That's our Calvin cycle pathway, our C3 pathway. The difference between C4 plants and cam plants is that cam plants open stromata at night, and that is when they fix CO2. They release CO2 in their inner cells and run the Calvin cycle during the day when the stromata are closed. So this is like a super efficient way to make sure that there's not too much oxygen around and they're not losing water. So basically CO2 is stored at night in cam plants and then it's used during the day for the Calvin cycle. Very well adapted plant to arid environments. So both C4 and CAM pathways function to concentrate the carbon dioxide to drive the Calvin cycle, to make it more efficient. C4 plants spatially separate carbon fixation and this Calvin cycle, and CAM plants temporarily separate carbon fixation and the Calvin cycle in a different time frame. So C4 plants divide these two steps spatially. They separate the carbon fixation, happens in one spot, Calvin cycle happens in another spot, and the CAM plants separate in time. They separate the carbon, they fix the carbon at night, and they run the Calvin cycle during the day. Now what happens to the sugar that's produced by photosynthesis? Well, that end product of the Calvin cycle, that G3P, that is going to be used to make glucose and fructose by the process of gluconeogenesis. We can also combine these molecules to form sucrose. When sucrose is really abundant, glucose is actually going to be polymerized to form starch. Starch is kind of plant's version of fat storage, which is a little bit longer term storage. Plants can also use starch and break it down at night into glucose to fuel their processes at night when photosynthesis isn't happening. So starch is often going to be found in the roots of plants to help them survive winter when photosynthesis isn't happening. All right, that is it, and that is the end of chapter 10.